Hi guys, I'm Dr. Rossler. I'm one of the biochemistry, I actually am the biochemistry professor here at Johnson & Wales, and so we're doing some online labs, and so my daughter, Zosha, do you want to swivel it to yourself? You just say hi. Who's 15, has helped me set up the labs. We're going to go ahead and do the labs that you would have done on Thursday and collect the data, and hopefully it works well so that you guys can go ahead and answer the questions and then still do the lab reports, all right? So hopefully Professor Lee has told you that this lab is on enzyme kinetics. So what you're looking at are enzymes, right? And enzymes are proteins that do jobs in the cell. They lower the energy of activation and they actually make things happen very fast. All right, and so the enzyme that we're going to be working on is one that's used in digestion. It's amylase. There's actually a couple forms of amylase. There's alpha amylase and beta amylase. And I think we're specifically looking at alpha amylase. It's in the saliva. And so my daughter Zosha has been nice enough to spit into a test tube multiple times. We're going to be testing her amylase, her alpha amylase activity. And so just to remind you what amylase does, and hopefully you remember this from other classes, is that amylase is going to break down amylose and amylopectin, which are the two components of starch. We're going to be looking at potato starch just because we have a lot of it sitting around. And so just to remind you what the structures of amylose and amylopectin are, is that they're made up of millions of little glucose monomers interconnected via dehydration reactions. And they have alpha-1,4 interactions as well as alpha-1,6 interactions. This may not mean anything to you right now, but it will as the quarter progresses. So here I made like a little pipe cleaner model of amylose or amylopectin. And again, just imagine that there's millions of glucose molecules all interconnected. So that's a lot of sugar. So what we're going to be doing in the lab to detect, what we're going to be looking at hopefully is how amylose or amylase, the enzyme that my daughter has produced in her saliva, is going to start breaking up and breaking down this large structure into smaller structures of actually maltose. So again, if this is made up of millions of glucose molecules, what we're going to see is that the enzyme Zosha produced is going to very quickly start breaking this down into, like this, into maltoses. And then the maltoses are later going to be cleaved down by maltase in the small intestine. As that happens, how are we going to detect that actually in this lab that we're doing today? So what we're going to be using is this is supposed to model iodine. So you guys might remember from Bio 1 labs that iodine has this affinity for starch. And the reason why it does is that what we're going to see is that the iodine atoms will actually very nicely fit in the alpha helices structure of amylose or amylopectin. So what happens when iodine is sitting here in these little, the little hole there, the little alpha hole of amylose or amylopectin, is that when it does this, you guys might remember that it makes a bluish color, like a bluish kind of purpley color, right? So what we're going to see in a second, and we're going to do this in a second, is we've got our starch solution, and we're going to add the iodine, and it should turn this sort of bluish purple color. As the reaction occurs, as we add the enzyme, let's think about what's going to happen. The enzyme is going to start cleaving more and more over time of that amylose amylopectin. And what we should see is that the iodine can no longer, there it is on the floor, the iodine is no longer going to be held in that hole. It's actually going to dissipate out, and no longer are we going to get the bluish color. So what we're going to see, hopefully, as the progression of the experiment occurs, is that the blue color is replaced by a clearish color as amylase does its job. All right, we can go ahead and pause it for now.